All right, let's finish this story off. And let me show you this paper that I wanted to show you about how an immune researcher called Peter Parham and his colleagues actually went to Africa and they sequenced KIR and HLA genes from 7 populations in sub-Saharan Africa. So you might know someone, you might be someone who is part of these population groups. I believe they went for isolated population groups so that they would have genetic distinctions among them. It's hard to find that, and they were looking for information about how can we see how these KIR and HLA genes are distributed among people, and how can we possibly use that to help us predict disease risk so that we can say things like these mothers are at risk for preeclampsia. That is what DNA testing can do. One important thing to say from the beginning, this is all about risk probabilities and populations. And so you can say that a certain mother is at risk for preeclampsia, but you can't say anything more than that. You can say that if they have this particular combination, that they will have a higher risk, but even the most reliable genetic screens still carry an 80% risk factor. That means that 1 out of 5 times, 20% of the time, that risk won't actually happen. And so we have things going on like that with this paper. So the thing I'd like to show you is I'd like to walk you through this paper. This paper, I believe, is freely available. Let me know if you need it, or you can always email Dr. Parham as well, because scientists are always happy to email you a copy of their paper. They don't want to keep it from people. It's the publishers that want to keep it behind paywalls. So just look at the title here. Diversity of killer cell Ig-like receptor, that is what KIR stands for. HLA class 1, which is the MHC gene locus, and their interactions in 7 populations of sub-Saharan Africans. These are the 7 populations, and also 3 other populations that they use the information from previous studies. One of the things about this, the 7 populations, they only were able to sample about 180 individuals. That means that you had maybe 13 in some populations, up to 50 people per population. You've got to be careful drawing conclusions from the results, because it's very possible that there are gene alleles and things out there that we don't know about. So you can't really draw conclusions about the populations from this, but you can draw conclusions about just the general spread of diseases and the association of disease risk. So these results are not definitive. I'm sure they would be the first ones to say that. The ones covered in this basically include from west to east the strip of sub-Saharan African tribes and populations. I believe that the ones, I'm trying to remember which ones of these, there were 7 of these that they collected new information for, but then they compared it to the other 3. Well, we can see around Burundi, there are a few. There's the Mbuti, there's the Hadza, there's the Datuga, and there's the Iraqw. And those 4 populations are going to show up in this study, and they're going to be compared to other ones from across the continent. So, a note about studying these. There's a lot of complicated numbers. You've already seen that the KIR receptors have really complicated names. That's because there's so much diversity. You can't give each one a simple name. You have to give it a complex name because there's a complex number of genes out there in immunity. The specific numbers are not part of the quiz, but there's 4 main themes that I would like to show you that come out from the data. And those, I'm actually going to summarize those at the end, and those are the things to go back and make sure that you study. The quiz will include how do KIR genes work, how do HLA, MHC genes work, and general conclusions about what they found in this study. Usually I'll put the most important information in the title of the slide, and when you're studying you should pay the most attention to that. Things like figure captions and small details of which tribe has higher of which allele, those do not matter to us. You can't even get that much useful information out of that level of specificity. What you can get is you can get general trends for diversity in KIR and HLA genes. So remember that each human will have 3 HLA class 1 molecules, A, B, and C, and you can be heterozygous for these. So when they looked at the 7 populations and they compared their 180 people, but I believe 10 had to be left out of the study for some reason. So out of 170 people that they were able to compare, 134 different versions, different alleles of HLA class molecules were found. They found 41 different HLA-As, 60 different HLA-Bs, and 33 different HLA-Cs. That already tells you right there, there's a lot of diversity. There's basically 1/3 as many alleles for an individual gene cluster as there are people. So probably most people have different alleles, because you can have a separate HLA-A from HLA-B and from HLA-C. So they looked across all 7 populations. None of the HLA-A or HLA-B alleles can be found in all 7 populations. But there are 3 HLA-Cs that are in all 7. This is because when we look at the role of the NK cell in immunity, the NK cell in immunity works with the HLA-C alleles. This other function keeps the HLA-C alleles more the same so that it can still interact with the NK receptors better. HLA-A and B are used more for infection than pregnancy, and therefore they have a lot more variation in them. So this trend has been observed before. We see it also with this study. HLA-C is less variable because it has a constant role to play in placental formation. So the other thing is, because look at this picture of the gene that's up here. You have the B and C genes on the chromosome, and then you have the A gene way over here. There's a large genetic distance between A and C. And so the thing is that because of this, the HLA-B and C genes tend to be the same in different people, whereas you will have more differences with HLA-A. We say that B and C are linked, and A is not linked as closely. For example, the Mbuti population had 5 different B and C alleles combined with the same A allele. So it looks like the B and C alleles could be switched off and recombined differently in different people. But the HLA-A allele, the B and Cs would go together, but the HLA-A would be different. The Mbuti also show evidence of a reciprocal combination event that's actually located between B and C. So sometimes you can have recombination that will cause variation in which B and which C a person has, but it happens less often. So the important thing is you have more variation in a single population for the A genes than you have for the B and C genes. The B and C tend to go together. Now in general, HLA diversity in Africa is high. They found a lot of those, that's a lot of genes. They looked at 170 people, and they found about 170 different genes when you add up the A, B, and C together. And so what they did to deal with this is they grouped the genes into groups called haplotypes. Now haplotypes are complicated, but it's important for you just to know it's a group of genes that tends to be seen more than once. So even with that, to try to simplify things, still, you had to have 204 different haplotypes to observe, that you observed in this study. Again, more haplotypes than you have people. 191 haplotypes were observed in only one population in the whole study. So that means only 13 haplotypes were actually shared by multiple populations. That's a small number, and that just says there's a lot of haplotypes, which means there's a lot of groups of HLA genes, a lot of diversity in Africa. It's one of the reasons why these studies are important to do in Africa, because there is so much genetic diversity that we can look at less diverse populations and not really understand all of the diverse combinations of receptors and genes that are out there. So the 13 shared haplotypes are only shared by a few people. This is a frequency, which is a number, it's a fraction out of 1.0. And so if you look at these numbers, they're really small. They're all 0.0 something numbers, which means they mean out of that population, for instance, for the Baka population, there's 0.28% of the population had this. That's actually one person. And honestly, I wish they had done that instead of doing it this way. I wish they had said one person had this haplotype in the Baka tribe, and then one person had the haplotype in the Mbuti. But they didn't, and if you look up in the Mbuti, you see that 6.3% of the people in the tribe had a different haplotype, haplotype 2, particular combination of A, C, and B. And that means 2 people had it. So these numbers are a little more complicated than they need to be, but you can see, you can look up and down, you can see these numbers are all small. The shared haplotypes are only shared by 1 or 2 people in each tribe, and there's only 13 overall. Out of 200 different haplotypes, only 13 are even shared. That means there's lots of diversity. There's lots of different HLA, A, B, and C genes in the population, which is good for immunity. It could be bad for pregnancy, however, because again, pregnancy is hardwired in the sense that you want to have a similar HLA to the KIR. For immunity, you want as much diversity as you can. So the fact is, despite all this diversity, they had a bunch of haplotypes, but all of the haplotypes contained alleles that had been seen before. This was the most surprising to the authors because they really expected, with the high amounts of genetic diversity, to be able to find at least a few new MHC HLA alleles. So they were expecting to find some, but it appears that HLA studies worldwide have already found all the major HLA alleles, which means that we probably know all of the HLA alleles that are in humanity as a whole across the world. And HLA genes are also diversified on every continent. Other genes aren't as diversified as HLA, but there appear to be pressures on the population to diversify your HLA genes, to have a lot of different MHC class 1 genes in the population. And this sort of makes sense because the population does depend on this to defend itself from immunity. Diversity was found. They did find some new alleles. They just had to look at the KIR locus to be able to do it. So on the NK cell side of the interaction, they found 28 new KIR alleles. So that's nice. They found something, and they got a better view of the kind of diversity that we can have in KIR molecules. They basically group them into 2 types of KIR haplotypes. They call it sen and tel, for closer to the centromere and closer to the telomere. And so for each of these haplotypes, you basically had 2 possibilities. You could have sen A or sen B, and you could have tel A or tel B. Realize that how they do the groupings is really complicated and statistical. What really matters is that you can do the groupings, and that you have 2 possibilities, A or B. And they tried to group them so that they could see what the diversity differences were among the KIR alleles. There's so much diversity among the KIR alleles that actually this is kind of hard to do, and it's statistically complicated. So there were 227 KIR alleles observed among the 200-ish or the 170 people that they looked at, 10% of which they had never seen before. Here's a breakdown of where the new KIR alleles were found. Here you have the 7 different tribes, and you have the number of sen A haplotypes versus sen B haplotypes for each one. Notice that each of these numbers, and then you have the tel A haplotypes versus the tel B haplotypes. Not everybody has to fall into a haplotype. Some of them, I believe, are non-haplotypes. So if you look over, you can see sen A and sen B, the total haplotypes add up to 180. But the tel A and tel B haplotypes do not add up to 180. That's just a consequence of the fact that these are groups of alleles. And realize that these are groups of alleles and what they call allotypes. The important thing is above my head, they will have the new alleles. And the new alleles that they found, every tribe except the Fulani gave them at least 1 new KIR allele. And the interesting thing that I see is that the tribes on the bottom of the table are the easternmost tribes, and those gave them the most new alleles. The Baka gave the most new alleles with 16. But then the Tatuga, Hadza, and Iraqw all gave above as many or more alleles than the other tribes that are farther distant. We're going to be talking about the Hadza tribes and the Iraqw later in this talk, and it turns out that this is part of what looks like an unusual immune system, okay, that they have. If we look over at this other part where we look at the KIR variants, the TLB, for these same tribes, and actually, so I'm going over here, you look at the new alleles. If you look at the bottom three, they have more than usual new alleles. They also have more than usual TLB haplotypes. That means whatever KIR receptors were put into the TLB group was unusually enriched in the Tatuga, Hadza, and Iraqw. Those are all Eastern Africa. And so it looks like we have some diversity in this area that we don't have in the Western Africa parts. As for the Sen-A and Sen-B, those look pretty constant. This is another graph that shows the same thing that I just said. And actually, if you look at this, if you look at TL-B versus TL-A, and you look at them and you arrange them in the eastward direction across the continent, which I believe this graph does, you can see that on the east side of the graph has more blue on the bottom, which is the teal genes, than the west side. There's no such distinction for the top of the graph, which is the Sen. So the teal genes looks to be like there's more diversity in these particular KIR genes, this particular grouping of alleles, in the eastern populations as opposed to the western. This is called a sign of directional selection. This has been looked at in other populations, and there's some reason that the environment will favor a particular allele. What they hypothesize is that this may show that immunity is prioritized in the West, and pregnancy completion is prioritized in the East. So the East may have more problems with eclampsia, but it may have fewer problems, but more problems with eclampsia, but the West may have more problems with malaria and viral threats. It's a very broad conclusion, and it's a question of priorities. Do you want to prioritize an immune system that fights off invaders, or doesn't fight off the baby, or plays its proper role in creating the placenta for the baby to grow in? Basically, fighting off something from outside versus having a successful pregnancy. The immune system is involved in both of those things, and it looks like there's some trade-offs that have to be in place during this. Different populations have different answers to these very difficult trade-off type questions. So it looks like TLB may prioritize the inhibitory immune reaction against the fetus so that the baby can grow. And of course, this is a mixture of inhibiting and activating KIR receptors. I'm just talking about having an immune system that can serve its role in the placenta versus having an immune system that can be activated quickly against infections like malaria and viruses. So remember, we had this slide before. There are 4 different alleles, A, B, C1, and C2, right? There's the A1, the B1, the C1, the C11, and the C21. So these 4 different epitopes are present on HLA, and these are what the KIR cells recognize. There aren't that many of them because you need to have them relatively constant. If they vary too much in a population, you have a much higher risk of having a KIR set in the mother that is inconsistent with the epitope set in the fetus. And in fact, there's some indication that immune system clashes may cause miscarriages and may cause premature pregnancies and things like that. So this is a graph where they look at the 4 different HLA epitopes, the 4 ones from that slide. A is yellow, B is green, C1 is pale red, and C2 is blue. Notice first of all that you have them organized by HLA A, B, and C. So these are the MHCs that contain those epitopes, and the epitopes are what will interact with the KIR. This is what needs to go with the KIR receptors, and if you have a mismatch between the epitopes on the HLA, the colors on the HLA, and the KIR receptors haplotype of the mother, this is what will cause the preeclampsia problem. So I want to walk you through this figure. It has the 7 tribes, and they're actually arranged roughly from west to east, with east now being on the bottom. But realize that we had the same thing we saw on the last slide. Well, actually, it's way back here. Remember that we saw more KIR variants, more of the KIR TL-B variants in the 3 tribes that were easternmost. So here, if you look at those are on the bottom of this graph, and 2 of those 3 tribes have distinctive colors. That means they have a distinctive pattern of HLA epitopes. If you look right over there, you can see that the boxed HLAs have a lot more green than the other ones above, and a lot less yellow. More of the B epitope, less of the yellow A epitope. This looks like part of the unusual immune system for the Iraqw and the Hadza populations. So notice this means down at the bottom, the other thing I want to point out is, hopefully I'm not covering it up, we have the HLA haplotypes, and this is differently color-coded. For this rightmost part, we have an indication of how many KIR ligands there are per HLA haplotype. How many HLA can react with the KIR. The most important thing is it looks like the same 2 populations have more KIR ligands per HLA haplotype. In other words, they have more KIR possibilities for reacting. That's shown on this slide. I actually moved it up and I showed you this slide itself. Remember here, yellow equals 1 KIR per HLA. Orange and red mean 2 to 3 KIRs per HLA. On the top of the graph, you have a lot of yellow. On the bottom of the graph, you have more orange. And in fact, in the 2 bottom populations, you have a majority orange. So you have more reactivity with KIR, more KIR per HLA that can actually react with the epitopes. Going along with that, you have more of the BW4 epitope and less of the A3 epitope. We don't know exactly what that means, but it's a pattern that shows that there may be disease risks that are unique to these particular populations. So this is what shows that exactly. The B4 epitopes are in green, the A epitopes are in yellow. You have more green for these 2 populations, and you also have more KIR ligands per HLA haplotype. It looks like this is a distinctive pattern of KIR HLA diversity, and so there may be distinctive patterns of natural killer cell related diseases, such as preeclampsia and pregnancy risks. The other side of things is that there are also distinctive patterns of immunity. We've got to remember these are patterns in the A3 and the BW4 epitopes, which are not specifically associated with the pregnancy part that we're talking about. And yet NK cells are involved in a very complex way. This is all very complex. Basically what I want to say, look at all that green. That green is distinctive. There is something distinctive about the number of MHC that the KIRs can bind in these 2 populations. And that's all we can say right now. We're looking for patterns and we don't know what they mean. So, to sum up, we're at the end of the paper. To sum up, Africans have a higher than average number of HLA KIR interactions relative to the rest of the world. If you add up the possible HLA KIR interactions, that means you see that all these numbers run from 6.8 to 8.4. In all the other non-African studies, all the other continents of the world, we got 6.5 or below for this number. That means the high genetic diversity of Africa results in a high diversity of HLA KIR interactions. So that makes sense. That's what we were expecting to find, And it also means that it's important to study the African genes because we don't understand yet what the diversity means. And so the best place to study diversity is where there's the most diversity, so you can possibly see the most interactions. Notice in all of these as well, HLA KIR interactions are related to what makes the Iraqw and Hadza distinctive as well. So it's possible that these are distinctive among the world for the diversity of KIR that they have. Again, we don't know what it means. It probably means certain risks for certain diseases and certain protections against certain diseases. So this is how it actually goes together with preeclampsia. If a population has a high proportion of that one inhibitory KIR receptor, that's shown in the first column, the proportion of that. If they have a high number in that column, or if they have a high number of the C2 HLA epitope, which will also cause the other side of the interaction that causes this. So it takes an interaction. You need both the inhibitory receptor and the C2 epitope to form this problem, to have the preeclampsia risk. And so what you do is you look at the population and you say what percentage of the population has this KIR receptor. And in the case of the Dogon, there are 38.6% of the population has the inhibitory receptor. 80.6% of the population has at least one of the C2 HLA epitope. You put those numbers together and you find out that there is a 31.2% risk for having a combination of the two of those. Actually, you take the numbers and you multiply them together. 38.6% times 0.80, which is the 80%, gives you the 31.2%. I believe that's exactly how these numbers work. So that results in the Dogon and the Fulani have a higher risk for preeclampsia. According to this one measure of preeclampsia, they are 1/3 of the population would be at risk for this. Now remember, there are other receptors that can compensate for this. This does not account for that. This is just saying, do you have the one problem? And so you got to remember these are probabilities and complex, but it does point out that there's a particular risk. Somebody working with the Dogon or Fulani population may want to check for signs of preeclampsia in their maternal patients. On the other hand, the Iraqw people have a 15% frequency of the inhibitory KIR receptor and only a 66% chance of the baby having the C2 epitope. Both of those are low, and they actually multiply out to give a 10.2% risk for preeclampsia. That population is protected against preeclampsia just by the fact that they don't have this particular interaction. You can see the C2 lines. I pulled the C2 lines for these three tribes. And so the Dogon and Fulani are on top. And you can see that the C2 epitope, which is blue, they definitely do have more of that. They have the C2, more of the C2 epitope, whereas the green box from the Iraqw will end up with a smaller amount of blue, and therefore a lower chance of having this particular epitope. If you have less of this epitope, you have less chance of this cause of preeclampsia. That's a pretty big difference, actually. If you look at those populations, you're talking about a 10% chance versus a 1/3, 33% chance for the other two. And that's significant. You know, 1 out of 10 patients is at risk from this particular thing versus 3 or 4 out of 10. And so that may affect what you see as a doctor. So it's important to be able to look at these kind of studies and to understand what they're saying, also understand they are extremely limited. But as we gather more information, we may become better at least identifying some major disease risks that can actually inform medical care. So this is my last slide. The main conclusions of this study are these 4 conclusions. There is high genetic diversity in Africa. There was high genetic diversity among the peoples that were studied. And yet, no new HLA alleles were found in any sub-Saharan populations. They take that as possibly concluding that all HLA genes are now known. This is good news because the more we know, the more we can associate with disease risk and prevention. many new KIR alleles were found. KIR has been less studied than HLA, for one thing. And this shows that we do need to keep searching for more new KIR genes. And the continent of Africa did provide the genetic database with more KIR genes that can help us understand disease better. So that argues that we should study the genetic diversity in Africa, can be beneficial to the health of Africans and the health of the world. KIR diversity does have that interesting change from west to east across the continent, which could possibly be a trade-off of disease, which is emphasized in the west, versus pregnancy success, which is emphasized in the east. This is very big, broad conclusion. This is probably a 1 to 2% actual real life effect, if it is that. So don't overplay this, but do realize that when we talk about disease risks and pregnancy risks, there can be genetic markers of risk that we can find out more about. And different populations will have from a low of 10% to a 36% risk of this particular kind of preeclampsia. So could a DNA test, if the day is around the corner where we could take a patient and we could actually sequence their HLA and sequence their fetal HLA, sequence their maternal KIR sequence, we could actually we could look for this particular combination of epitope and of fetal epitope and maternal receptor. And that could diagnose, we could say to that mother that you're at risk of preeclampsia. Even without a DNA test, we can start to think about what cases should a doctor be more vigilant for particular diseases. Lots of questions with how do we apply this knowledge. All I'm really doing here is scratching the surface. And I want to show you that this knowledge is here. This comes from the DNA mechanisms that we have, and it comes from understanding how proteins bind proteins. And that's what my area is. So I'd like to bring that to you and let you know that this is possible, and I hope that you can be aware of it now. So learn about this, learn about these 4 main conclusions, and that's what I hope will help you as you move forward to becoming doctors and serving your patients in the best way possible. All right. Well, I've had a long day, so I'm going to go to bed, but maybe we'll have a Q&A sometime soon. My recording here is done. Good night.